Hello everyone, and a really warm welcome to this session, What's on the Horizon for Ovarian Cancer Diagnosis and Treatment. I'm Lizzie Hussain, I'm Deputy Director of Services at Target Ovarian Cancer, and I'm delighted to invite our panellists, Professor Usha Menon and Professor Charlie Gourley, to share their perspectives with us today. Anything that comes out of this session isn't medical advice, and it isn't a substitute for the information or advice that you've been given by your own clinical team. And so without further ado, let's get started. Thanks, Lizzie. Thanks very much for that, that introduction and for the, invitation to talk today. And what I'm going to talk about is translating research into clinical practice, because that's essentially what we do. So what our team here does, and what many teams around the country do, is that we try and go from the bench, the lab, to the bedside and back again, actually. So things that we learn in clinical trials, we try and work out, why we get the results we do by laboratory approaches. And those approaches are to look at cancer cells grown in the lab, to look at their basic biology, to look at why they've become resistant to some of the therapies. We also take clinical material, so patients' tumors, and we analyze them. And largely speaking, that means we look at the DNA or the RNA or the protein, which I'm going to talk to you a bit about in a minute. And then the other thing we do is we do clinical trials. So we compare new drugs versus our standard of care drugs. And if we see improvements, we try and work out which patients benefit most and what to do in the patients who don't benefit so much. So this DNA to RNA to protein thing, some of you will know about it, some of you won't, but this is a sort of, I guess a description of how it works. The DNA is the cookbook. So if you imagine it like cooking a cake, then the DNA is the cookbook and the RNA is a copy of the recipe. And the cookbook and the copy of the recipe are all in the library, which is the nucleus of the cell. But then the recipe gets taken out into the kitchen, where, according to the sequence of the RNA, amino acids, which are the building blocks of proteins, are put together to make this cake, which is the proteins. And it's proteins that make us all up. They make, they're the building blocks of our body. And so what happens in cancer is you get mistakes in the DNA, which then get translated into mistakes in the RNA and therefore mistakes in the protein. And how we investigate this is, well, we can look at the DNA sequence, or we can look at the RNA sequence, or we can look at how much RNA there is for a gene, i.e. how much it's switched on. Or we can look at the protein, how much protein there is. And this figure on the right-hand side is a sample of a tumor that's been stained, and the tumor cells are stained in brown for this protein, so we can see how much protein is produced, or how much of this particular protein is present in these cancer cells. And this is how we can dissect what's been going on in the tumors. And the thing is, I guess when I started doing this about 15 or 20 years ago, we would look at the DNA of one gene and see what that meant, or the RNA, one RNA, or one protein. But now we've gone to looking at every gene. In fact, we've gone beyond that. We've gone to looking at bits of the DNA that don't even encode genes, because it turns out they're also important. And then we've taken to looking at the whole RNA that's produced in the cell and analyzing that and looking at networks of proteins. So we've gone from this situation where we've got a small amount of data to an enormous amount of data. And we've got to somehow make sense of all of that. And we're very lucky here in Edinburgh. This is our institute, and the big fancy looking building at the front there was only built about 5 or 6 years ago. And it was built to join the 3 buildings behind it together. That was one of the purposes. The other purpose was to give us 2 floors worth of bioinformaticians. So those 2 floors of that building that you see are full of bioinformaticians. And these are clever sort of computer scientists that crunch through data of sequence data or gene expression data. And we've got 120 of these here that help us analyze all our data. And it's been incredible actually having these. They've really helped us. And I guess I just thought this would be a good point to also mention that in terms of all of this research, the number of different occupations that are involved, you've got radiologists, geneticists, various statisticians, surgeons, nurses, radiotherapists, physicists. I mean, it's a massive team, I guess, and that's why it's good to work in a place where you have all these members of the team that you can bring together. So in terms of the approach to ovarian cancer, I'm very much a believer that we don't treat it as one disease. We have to treat it as multiple different diseases. And the first way we can split it up is according to what the pathologist sees when he looks at the tumor down the microscope. He or she looks at the tumor down the microscope. And so we can split it into 5 main types: mucinous, endometrioid, clear cell, high-grade serous, and low-grade serous. And the thing is, these all behave very differently. And so, they respond differently to chemo, they have different outlooks, they respond differently to different molecular therapies. And so it's really important to split them up. And I'm going to just give an example of 2 of them, high-grade serous and low-grade serous. I mean, we are working on them all actually, but for reasons of time, I'm just going to talk about high-grade serous and low-grade serous. So if we talk about high-grade serous ovarian cancer, well, it turns out that it has an Achilles heel. What that is it's got a weakness that we can exploit. So this kind of complicated looking figure here shows changes in the number of copies of genes in various different cancers. And it turns out that the cancer that's got the greatest number of changes in copies of the genes is high-grade serous ovarian cancer at the bottom there. There are more big red bars and more big blue bars than there is in any other of the cancers. GBM, which is a brain cancer, also has quite a lot of changes, but ovarian cancer, the reason it's got all these changes is because it's got defects in its ability to repair DNA. And it turns out that this difficulty it's got in repairing DNA can be exploited, and we can push it over the edge using a drug called a Parp inhibitor, or a group of drugs. There are a number of them now. And it's only really been over the last 5 to 10 years that we've realized, how much of a game changer they are. And so this figure on the left-hand side is from a study in relapsed high-grade serous ovarian cancer. And what it shows is that out at 3, 4, 5, 6 years, there are quite a lot of patients who remain on this therapy. And relapsed high-grade serous ovarian cancer is something that we used to think was not curable. But in some cases, and I'd hasten to add, unfortunately, not all cases, but in some cases, there are patients now who had relapsed ovarian cancer who remain in remission 10, 11, 12 years down the line because of these new drugs. And so that's now led to us putting them into the first-line setting. And if you look at the figure on the right-hand side there, you will see this is what's called a survival curve, or a progression-free survival curve. It tells us the percentage of patients whose disease has not progressed. And this is from a fairly recently done study called SOLO-1, which gave patients the Parp inhibitor olaparib after their first-line surgery and chemo. And what this shows is that out at 5 years, almost half the patients have not progressed if they got the olaparib. The patients on the control arm, 80% of them have progressed. And this is really a result that we've never seen the likes of before. So it really is a game changer. All the patients in this study had BRCA1 or BRCA2 mutations, and that's only 20% of all high-grade serous ovarian cancer. And we now know that more patients than that benefit from this sort of therapy. So what we've got to try and do is work out, who are these other patients that do benefit? And one of the things we've been doing here in Edinburgh is we've been doing quite sophisticated sequencing of our high-grade serous ovarian cancer patients. So we're doing this thing called whole genome sequencing. And when we do that, one of the first things that came out of this study was that we found we could identify an additional 15% of patients over and above what was detected by conventional sequencing that had changes in their BRCA1 and BRCA2 genes, and so therefore may benefit from this, from Parp inhibitors. And this is a collaboration that's across Scotland. And it's also allowed us to look at other of the secrets of the cancer genome. It's shown some incredible, complexity of the genome. This figure at the top in the middle here shows huge complexity of some of the genomes. It shows that we can get two genes fusing together that can actually have an important role in the cancer. It also shows that there are bits of DNA that come out of the chromosomes and then multiply themselves. And we think that's also important. So we're still in the process of analyzing this. And we've currently got about 8 bioinformaticians working on this data. And they have been for the last 3 years or so, because it's a real complex problem for them to crunch through. But we think it's uncovering some of the secrets for us. So hopes for the future in high-grade serous ovarian cancer. Now, I think in the short term, our best chances of making progress are to exploit these problems that high-grade serous ovarian cancer has in DNA repair. And I've just mentioned 3 drug classes here: ATR inhibitors, WEE1 inhibitors, and something called DNA polymerase theta inhibitors. And I think in the short term, I'm quite excited about what they might achieve. Now, you've probably heard about immunotherapy, because immunotherapy works well in other cancers. Unfortunately, the immunotherapy drugs that are currently available don't work fantastically well in high-grade serous ovarian cancer as yet. However, there are many different strategies being employed. And I think in the long term, immunotherapy may well be important. It may require different drugs, different approaches, perhaps putting some immune cells back into the patient. So that's something to look out for in the longer term. So if I turn now to low-grade serous ovarian cancer, and I'm going to tell you about a study that we were heavily involved in. We were the UK leads for this study that happened in the UK and the USA. And because low-grade serous ovarian cancer, it's a cancer that's driven by a pathway in the cell called the MAP kinase pathway. And this is a pathway whereby the cell gets a signal that goes from the outside into the inside and tells the nucleus to divide the cell, for the cell to move, for the cell to potentially metastasize. And it happens because either the RAS or the RAF gene has a mutation that tells the cell to continue this signal even when it's not getting a signal from the outside of the cell. But what we can do is we can block that signal with a drug called tremetonip, because it inhibits this MEK protein here. And so we did a study comparing tremetonip to standard of care therapy in patients whose low-grade serous ovarian cancer had come back. And when we did this, what we showed was that the rate at which the cancer came back was halved when you used tremetonip compared to using the standard treatment. Also, the percentage of patients who responded to tremetonip was 4 times higher than the response rate in standard of care therapies. So that's great. It's a really good result that we're very happy with. This paper is about to be published in the Lancet, and it's led to this drug being recommended in the recommendations in the US and the UK for treating this disease. It was also the first ever positive randomized trial in low-grade serous ovarian cancer. And what we're doing in Edinburgh is we're now sequencing the tumors of all these patients in order to try and work out which patients respond best and which patients maybe don't respond, and we need to find a different strategy for. So the next steps for low-grade serous ovarian cancer. So as I said, we're going to try and work out using the genetics or the genomics which patients are most likely to respond. We're also doing a drug screen of low-grade serous ovarian cancer cells in the lab in order to try and identify new drugs to treat low-grade serous ovarian cancer with. And anything that we find, we will try and put straight into trials. And this is being done by one of my postdocs, Rob Hollis, who's pictured here on the right. And this bit is actually funded by Target Ovarian Cancer. So we're very grateful to them for that. It's also important we start new trials, and there are exciting new trials starting up. There's one recently led from the Marsden that's being conducted in some of the big centres around the UK, and we're also helping with that. international collaboration is crucial because this is quite a rare form of ovarian cancer. And so working together is really crucial. And what we've also got to try and do is to think outside of the box. And that's why the picture of this avatar is here, or from the film Avatar. And the reason is that I think there may be scope in this disease to try and take samples and maybe grow them outside the body and try and work out what drugs work best in each patient, and try and adapt what drugs we give to each patient according to these models. And this is something I'd really like to do in low-grade serous ovarian cancer. We've not initiated this as yet, but it is something that I would really like to start doing. And I think we'll just skip this one for reasons of time. But this is my last sort of factual slide, and it's just showing new drugs that have been approved over time to treat ovarian cancer. And if you go back, if you notice in the 1960s and the 1970s, there were only 1 or 2 new drugs every decade. And even in between 2000 and 2010, there were well, really just 2 in that decade. And now look at it, between 2010 and now, there are a huge number of new drugs. So I think, you should bear that in mind, that the progress is now accelerating immensely. And these drugs are obviously only approved after clinical trials, and it shows the value of clinical trials. So in summary, personalization of care is crucial to maximize the utility of new ovarian cancer drugs. And it's also really important to facilitate the discovery of new drugs, as well as maximizing the use of the new ones that we're testing. And as we become better at harnessing the big data, whether that be sequence data or imaging, this will help us better understand what drives the cancer vulnerabilities for each of these specific cancer, for each patient's cancer. What is their Achilles heel? And do they have cells that make them resistant to drugs? And can we do something to prevent that resistance? So the progress is accelerating, and clinical trials are key to proving that our stepwise changes in therapy are also improving patient outcome. So I think the future is bright. I think it feels as though we are accelerating our progress. So I would feel very positive about all of that. I would just like to acknowledge everybody who contributed to the LOG study, which is the low-grade serous study that I described, and also everybody who helped with this Scottish sequencing project that I also described. And then also to mention our NHS infrastructure, which helps us a lot. And also in each of these acknowledgements, we must remember the patients who either donated the samples or who participated in the study. Okay, thank you very much. Thanks for your attention. I will take any questions.
Thank you, Charlie, for such an interesting presentation. As Charlie mentioned, we have time now for some questions. We've received a few questions in advance, but we also should have some time to answer your questions if you pop them in the chat box. We might not be able to get to everybody's questions, but we will try and get through a few. So let's get started. Charlie, I'm going to start you actually with a question from the chat that somebody's asked today. So somebody's asked us, could Charlie please talk a little more about immunotherapy and perhaps explain why it doesn't work as well in ovarian cancer? How long might it be until we start seeing clinical trials?
So, well, there are clinical trials. And so what happened was immunotherapy appeared on the scene and actually works amazingly well in melanoma and some lung cancers and some kidney cancers. And it turns out that the cancers that it works best in are the cancers that generate a high number of what we call, well, they have a lot of mutations in them. So they're heavily driven by mutations. Now, high-grade serous ovarian cancer is not really driven by mutations. It's driven by changes in copies of the gene, numbers of copies. But these other cancers are driven by a lot of mutations. What that means is that the surface of the cell, the proteins are different from the proteins at the surface of the non-cancer cells. And for that reason, they're more immunogenic. And so they respond better to immunotherapy. And so we know that the number of mutations in high-grade serous ovarian cancer is kind of below the threshold that you need to get a good response to immunotherapy. It doesn't mean nobody will respond, because there are certainly reports of people who do respond. But in general, the number of responses in high-grade serous ovarian cancer is less. There is a feeling that in clear cell ovarian cancer, there is sensitivity, and there is a trial that has been done and completed, and we're expecting results from in the next year. So that'll be interesting. And I think, you know, the other issue is that it seems that some ovarian cancers are able to keep the immune cells outside the cancer, so they can't even get in to interact with the cancer cells. And so we're busy trying to find strategies to deal with that as well. So, you know, but, you know, I don't think we should feel down because ovarian cancer doesn't respond quite as well as other cancers to immunotherapies, because we have other weapons. And so other cancers don't respond as well to Parp inhibitors as ovarian cancers do, for example. So we've got targets in form of these DNA repair targeting agents. And we just need to explore those more, I think.
Lovely. Thank you ever so much for answering that, Charlie. So on to the next question. We've had a question about personalised medicine, which I think you touched on a little bit in your talk, but we'd be interested to hear your thoughts on how important you think personalised medicine will be for the future, and just explaining a bit about what exactly personalised medicine is and why we call it that.
Yeah, no, I think personalised medicine really is everything in ovarian cancer. So in the rare subtypes of ovarian cancer, so clear cell, low-grade serous, mucinous, they don't respond all that well to chemo, but they have molecular abnormalities that means they may well start responding to or they may well respond to other targeted therapies. And actually, I skipped over a slide in the talk because I was running out of time, but there is a trial that's about to start, that has started actually, because we're just putting patients in it now, that in these rare sorts of ovarian cancer, the strategy is to sequence the tumor and work out which abnormality it is. And then on the basis of that abnormality, they will get one of 4 different treatments. So that trial is driven entirely by the molecular biology or the genetic changes in that tumor. And in high-grade serous, I think it's absolutely crucial. As I said to you, there's no question there are secrets in the genome that we're just beginning to unravel. And already there are drugs like, so Parp inhibitors, BRCA1 and BRCA2 patients, you've got to try and get them a Parp inhibitor if they've got stage 3 or stage 4 disease. But we know beyond that, there are patients who respond to Parp inhibitors. And at the minute, there's a suggestion that there's a special genetic test that will pick them out. And that special genetic test is quite good. It's called an HRD test, but we can do a lot better. And so I think in ovarian cancer, the personalization issue is just going to get more and more important as we learn more of the secrets.
Thanks, Charlie. We had a question related, I think, to HRD testing. So someone's asked us, are there any developments in testing tumours in the UK? Could you perhaps elaborate on that a little bit?
For HRD, so HRD testing is coming to the UK. I believe in England, and obviously I don't work in England, but I believe this is something that is going to be available very shortly, because it's necessary in order to access a combination of drugs, olaparib and bevacizumab, and it is going to be driven forward. There are these things called genomic hubs in England, and there's going to be, it's definitely going to happen. I know that because I've had discussions, although I don't work there. And we're currently in discussion here in Scotland about how to do something equivalent. So it's very much, it's on the horizon. In fact, it will be here, imminently. Yeah, it's happening.
Thank you. So we've had a couple of questions about mucinous ovarian cancer. So people are interested to hear whether you know of any work going on or research into mucinous ovarian cancer.
Yeah, well, so we were involved in a big sequencing study that was done. It was actually led from Australia, and it was published in 2019. And that shed quite a lot of light on mucinous ovarian cancer, because there used to be a bit of a debate as to whether it was actually even a real thing and whether these cancers actually came from the gastrointestinal tract rather than from the ovary. But it became clear from that sequencing study that some of them probably do come from the ovary. And so, but what that sequencing study also showed is it also showed what were the key genes that would drive that. And by that, I don't mean inherited genes. I mean genes that, you know, that can be abnormal in the tumor. And the key ones, I guess, are KRAS and HER2. And so, so now there's this study that, now I'm really regretting flicking over that slide, but this study called Bouquet, which is just starting. I think it's being done in one of the centres in London and here in Edinburgh. And this study, patients with mucinous ovarian cancers will get their tumor sequenced, and then if they have one of these mutations, they will go in to the treatment according to the mutation. And actually, in the study, if the patient doesn't have any of the mutations that would put them into one of the 3 mutation-driven arms, then they go into a 4th arm that's got an anti-angiogenic drug and an immunotherapy drug. So, you know, there is potential for anybody with a mucinous tumor, as long as they fit the inclusion criteria, to be considered for that study. So that, I think that is the only mucinous study I know of in the UK, but you know, I'm sure the people in London, it's at UCL, I'm sure they are taking referrals, and we are certainly taking referrals here in Edinburgh for that study.
Thank you. And just to add to that, we've been asked if there are any advances that you could talk about in terms of treatment for other rarer cancers, such as granulosa cell tumour. So,
yes, so granulosa cell tumours, I mean, there has been some research. I have to say, there hasn't been, at this point in time, kind of earth-shattering change, because it's quite difficult to do studies in those really quite rare groups. There is a group, the GCIG, which is the international group that kind of oversees all of gynecological cancer research, who have a rare cancers working group, and that group is putting together some granulosa cell studies with international collaboration. You know, but it's certainly somewhere that needs more work, and yeah, I'm sorry that there isn't more in that area.
Thank you. We're going to circle back a little bit now. We've had some questions about PARP inhibitors. Yep. So I think you mentioned in your talk that we know that people with BRCA mutations benefit from PARP inhibitors, but we've had a question. Could you explain who might benefit from PARP inhibitors in addition to those with BRCA 1 and 2 mutations? Could it be anyone with high-grade serous ovarian cancer?
Yeah, it could be. So essentially the way it works is that anybody who has responded to their chemotherapy could benefit from a PARP inhibitor. The chance of benefit kind of shifts in a stepwise fashion, such that patients with BRCA 1 and BRCA 2 mutations, they've got a very high chance of benefit. And then patients who don't have a BRCA 1 and BRCA 2 mutation, but are positive for this HRD test, then they also have a pretty good chance of benefit. And then outside of that, there is the group whose HRD test is negative. And actually, but some of them still do benefit. So there were 2 trials that looked at this. One of the trials, the patients got bevacizumab in the run-up to the PARP inhibitor, and the HRD negative patients didn't seem to benefit in that study. But in the other study, the patients didn't get bevacizumab on the run-up to the PARP inhibitor. And even the HRD negative patients did get some benefit. So this is where we've got a slight difficulty with the tests. We still really are not entirely sure which patients will definitely benefit, and more importantly, which ones definitely won't benefit. So I think as long as patients have responded to their chemotherapy, there's a chance they will benefit, I would say, from a PARP inhibitor.
Thank you. And kind of building on that, we've had a question about PARP inhibitor resistance. So somebody's asked, is there any information on PARP inhibitor resistance and how that could be overcome?
Yeah, so that's a really hot topic, actually, because, you know, we want to know why patients become resistant to PARP inhibitors, because what PARP inhibitors provide is clearly such an amazing benefit. And so if we could work out what it was that caused patients to become resistant, then maybe we could change what therapy we gave them before their PARP inhibitor, or what surgery we gave them before their PARP inhibitor, or maybe we could find drugs that would block that PARP inhibitor resistance. And so we're beginning to get some idea of what the PARP inhibitor resistance mechanisms are. And some of them revolve around getting a further mutation in the gene that's driving the ovarian cancer. So say it was a BRCA1 gene that was abnormal, then patients might get another change in that gene, which then makes them resistant to the PARP inhibitor. But there are other mechanisms as well. And the slight difficulty is we don't know the proportion of those mechanisms. So others and us, we're currently starting a study in collaboration with Glasgow, being led from Glasgow, but and my lab team are doing a lot of the analysis, where we're taking 200 patients who've got samples from before and after their PARP inhibitor to try and work out what all the mechanisms are. And the reason we're doing that is there are now drugs that we think might block some of these mechanisms. So it's early days, but there are a lot of people working on it, and we're working on it as well.
Thank you. So we're going to go back to low-grade serous ovarian cancer now. So someone's asked, you mentioned the outside-the-box approach for low-grade ovarian serous that you'd like to do. Is there anything planned for this at all, or do you think it's something that is likely within the next 2 years, 5 years, 10 years, and what would that be dependent on?
So yeah, so I'm hoping 5 years, and what that depends on is our ability to demonstrate that we can kind of grow and characterize the tumors outside the patient's body in a way that is meaningful. So, you know, I mean, for 20 years or so, people have tried to grow the tumors outside the body and use that to predict sensitivity to drugs. And, you know, 10, 20 years ago, it didn't seem to work very well. However, our ability to grow cells outside the body in these things called organoids and grow them in 3 dimensions is much better than it used to be. So what we need to do before we can do such a study is demonstrate that we can grow the cells outside the body in a time frame that is not so slow that it can't guide treatment in the patient. And this is why I think low-grade serous ovarian cancer is the perfect cancer in which to try this, because low-grade serous ovarian cancer generally is a slow-growing cancer. So you would have time to do the lab analysis at each step of the way, and then for it to inform what therapy you were to give, hopefully in most cases. So that's what we're currently doing just now, is refining that as well as improving our molecular characterization, which we're doing. Yeah.
Thank you. So on to a slightly more general question now. So we all know that research has been impacted by the pandemic. What would you say were the other key challenges for ovarian cancer research, and how would we work together to overcome them?
Okay, well, so yes, I mean, the pandemic's had some impact. It's had probably more impact on the lab rather than, so it was certainly in Edinburgh, we were able to get our trials up and going again quite quickly. But the lab was affected significantly. And so, people doing PhDs, etc., the research was knocked back around 6 months. And so that's been a big issue. I think the biggest issue that we would like to overcome is to mainstream genetic sequencing, complex genetic sequencing for our patients. So, you know, 10 years ago, we weren't doing routine BRCA1 and BRCA2 sequencing for our ovarian cancer patients. And then in 2012, we started to be able to get that in Scotland. And then it started around the same time in England as well. And now we do, you know, for our ovarian cancer patients, we do a panel of 8 genes here. And I think they do something similar down south. But what I want to move to is a situation where we're sequencing all the patients' genes for all our patients. And that is quite expensive. It might cost £1,000 or £2,000, but the drugs we're giving to patients cost much more than that. So I think we can justify it on the basis of making sure we give the right drug to the right patient. So I think that's the biggest challenge, is being able to make the argument that we do that mainstream, because as soon as we start doing that mainstream, it then gives us all this data to look back at what happened to our patients and to try and predict what drugs will respond best to. I think it will open huge avenues, and it will also make drug companies want to come in and work with us with their new drugs.
Thank you so much to all of you for joining us. We hope you enjoyed it, and we hope you found it as informative as the team did as we move forwards together. I'm really sorry if we weren't able to answer your specific question in this session, but please do remember, if you have any specific questions or concerns, or you'd just like to have a chat, our support line is available, and I've spotted lots of questions that we would be able to answer on our support line.